All right. I think we are live. And Paul, would you like to kind of take it away?
I sure would. Thank you, Vikram, for that. Let's see here. So today, here we go. I am going to cover PGRs, plant growth regulators for ornamental crops. And this is just going to be an overview of what's out there, what's available, how they work, and some background information on them. So if you're a grower, whether it's nursery, greenhouse, there are plant growth regulators available. They are a tool that you can use. And over the years, these options of plant growth regulators have expanded. One of the reasons is a lot of the active ingredients that are used in these plant growth regulators are now off patent. So there are a lot of other brands out there. And just be aware that some of the AIs, the concentrations may vary in the concentrate product from brand to brand. So just be aware of that. If you're using one at one concentration and it's a different uh, concentrate that's available, you may run into some issues. So as we say with everything, make sure you're reading the labels and being aware of what you're working with. How do they work? What's their roles? We want to control growth. And why do we want to do that? So these chemicals, active ingredients, they affect GA, gibberellic acid synthesis, which influences cell elongation, which causes the plant to grow and get taller. And so these chemicals, most of them, there are some that do other things, they affect that growth. They stop that growth and keep that plant more compact. And one of the reasons why growers like to do that is by controlling that plant growth, they can grow more plants per bench. Shipping is easier. We can fit more plants on a rack. Um, so that helps out. When it gets to the garden center or the nursery, the product's easier to handle. It's in blooming, it's uniform, so there's a lot of pluses when it's done correctly, and you're using the proper PGR at the proper rate at the proper time. You're going to have a high quality product. But they can also cause flower bud abscission. All right, so we could cause buds to abort or drop. It helps increase branching. So instead of coming through and pruning, some of these can be used as almost a chemical type pruning to increase the branching. They do help promote flowering. If we've got more branching, we've got more potential spots for flowers, buds to develop. And in certain cases and in certain plants, we need to stimulate shoot elongation. So there are some products out there that have GA and also benzyladenine in them. So, and we'll touch on them as we work our way through. As I said, make sure you're reading the label. Specific recommended dose ranges are on there. Some of these can be used as drenches. Some of them can be used as foliar sprays. So make sure you are um, well aware and familiar with that. We talked about application recommendations, whether it's a drench or a foliar. And then, precautions there. Some of these do have some phytotoxic effects. Even if they are applied properly, you might see some yellowing or you might see some, minor side effects. So just be aware of that. So if that crop is getting close to market time, you may not want to do that application because it may have some negative effects on the overall sellability. So let's talk about get into them. So the first one is Ancemidol. Out there, two examples are Abide and A-Rest. It moves readily through the plant, which is nice. So it gets to the point of where it has to interact with those cells that are dividing and elongating. It's used on crops mostly where the other chemicals, the other PGRs aren't effective, and often on very high value crops such as bulbs. Sometimes we run into issues with bulbs. We need a PGR to slow them back or hold them back, whether it's due to climatic conditions or something. So Ancemidol works extremely well. Plug growers really like it because of uh, it has a reduced risk of phytotoxicity. They give that name, it's a safer PGR to apply because it has that limited residual activity. So what that means is as that plug is transplanted into its final growing pot, it grows out of those conditions nicely, and it doesn't have a delay or negative effect on the overall growth response. So that is Ancemidol. Next, we've got Chlormyquat, Psychocell, which has been around forever, and it's been used on everything from poinsettias, geraniums, osteos, hibiscus, you name it. This works much better as a foliar application, but this is one where you will see some yellowing, especially on some of the older foliage. When it hits it. So again, make sure you have enough time for some new growth to flush above it, and it will grow out of that yellowing. Chlormyquat does extremely well in tank mixes with diminizide or B9. So we get better height control. We reduce the potential for phytotoxicity because we're using a lower concentration of the Chlormyquat. And I would not recommend this tank mix as a drench because it is not cost effective, and B9 does not work well as a drench. In fact, it's never recommended as a drench. So Chlormyquat is another one. You can see some of the examples here of with B9 and Psychocell, how they respond. Citadel is a brand name for Chlormyquat, and Dazide and B9 are the brand names for the diminizide. So you can see their responses there, similar in response. So this is good to know that these, depending on the brand that you prefer, who you work with, you're going to get similar type of results. All right, diminizide or brand names are Dazide or B9. Apply it only as a spray, and it is highly mobile. So it gets to those growing points, moves quite well within the plant, taken in by the foliage and in through the stems. Very effective on just about every crop except the lilies. And so this is where Ancemidol comes in. You would use that instead of using the B9. It's also effective in controlling seedlings in the plug flats, so it works very well there. And it's more effective, or it's most effective in cooler climates. So this time of year, I'm in Houston. If you're a grower, you're probably not going to want to use B9 because you're probably not going to get the results. Now, in certain cases, and if you know your crops and if it works well, by all means, but as a rule of thumb, it's not going to work as well in the hotter temperatures. So this time of year, you might want to consider one of the other PGRs in order to get the quality or the response that you want. Dikegulac sodium, Atromec. This is registered for greenhouse and nursery. There used to be another product out called Agio, which is no longer on the market, and it was a similar type active ingredient, same active ingredient. And this one temporarily stops the shoot elongation and promotes lateral branching. So when I was on the nursery side of things, we would use this as a type of pinching agent, and we would get very good responses to it. It would stop that that elongation, hold that apical growth, and then we would see very nice branching coming out along the stem. So it's a retardant pinching agent. There's a couple species there that it works on: azaleas, bougainvillea, clerodendron, lantana, and verbena. And just be, like anything else, if it's the first time that you're going to trial one of these or use one of these, bracket your concentrations and just try it on a small test plot before you settle on the concentration that you like and you get the response that you want. This Atromec or the Dikegulac at higher rates can really cause some heavy stunting, and the plants it will take a while for them to grow out of it. So, and this could be the case with any of these PGRs, a little is good and more is better, and that is not always the case. Follow the label, follow the concentrations, work with your reps that have the experience, or talk with the technical people from the chemical company in order to make sure you're in the right area with the concentrations. Ethaphon, phosphoric acid, has another one that's been around forever. Florel or Colate are two of the brands that are out there. And this one works a little differently. So this one is not affecting GA or gibberellic synthesis. This one, the product, the Ethaphon phosphoric acid, is absorbed by the plant tissue, and it changes the pH in those cells once it's absorbed. And then ethylene is released from these cells and acts as a promotes the flower bud abortion and enhances vegetative branching. So as you can see in this image here, our control with geranium, you can see non-treated, the number of flowers, we can probably see about 3 or 4 buds there at 500 parts per million Florel. You've got some nice growth control, but the number of flower buds that are coming is dramatically increased. And as we can see, 1,000 is probably on the high end, too much stunting and not enough bud break. So rule of thumb for Florel is usually in that 250 to 500 parts per million range for applications. Fluoropyrimidinol or top floor. This one is closely related in how it works to Ancemidol, which was the first one that I had talked about, but it has a greater degree of activity, so it's a little bit more active, and it's very cost effective as a drench. You can see here in these this image on the concentration on these drenches on these poinsettias. We're at 0.05 parts per million, 0.1, and then 0.15, and you can see how it compares to the tank mix of Dazide, which is similar to B9, and Citadel, which is the Chlormyquat. So you can see very low concentrations as a drench, and you can see the height control, and then the flower production that you get. Little decrease in the overall bracts size as they mature, but the number of bracts and the number of flowers that are there is definitely increased. For containerized growers, for nursery growers, this is one you might want to look at because there's also a granular formulation. So if there's a couple plants, some species that you might be having issues with in getting away for you, or you just want to control them a little bit more to get a better overall look, you might want to consider looking at the granular formulation of top floor to get those plants under control. Paclobutrazole, again, another one that's been around for quite a while, and you can see there's several out there: Piccolo, Bonsai, Downsize, Pac-O. This one is probably the most widely used in the greenhouse industry for floriculture crops. It is effective as either a foliar spray or a drench. So this is one where we can see how nicely it works. This is the Bonsai formulations at 1 part per million, and then this is at 2 parts per million. So you can see, compared to the control, very nice response to it. That 2 parts per million control is really nice. That will ship much better. This on a rack with multiple plants on there, we're going to see some sort of damage. You may have to sleeve something like this on the far end. At the 2 parts per million is going to pack nicely on the racks and with a lot less damage and a much better quality when it gets onto the benches at the garden center. Uniconazole is a newer one, I would say. It's been around a while, but here is Concise and Sue Magic. This can be used as a foliar spray, a drench, or even a liner soak. But the one good thing about it, when we compare it to Paclobutrazole and the Chlormyquat, we can use much lower rates as a drench compared to these two, and it's often used on perennials. So in these are New Guinea impatiens. You can see the 5 parts per million on the two above, but down at euconazole, we can get by with half of that concentration. And you can see very low concentration is going to give you the response that you need. And at least in this case, at 5, 10, and 20, that plant is way too stunted. So always, consider that, it's not just about the ease of use. Make sure you're putting pencil to paper and running the numbers to see which ones are most cost effective also and make sense for your operation. So now we're talking about these non-GA inhibitors. Benzyladenine, Configure is the brand that's out there. When I was doing tissue culture running the lab, BA was benzyladenine is always used for shoot proliferation for cultures. And so now on the nursery side and greenhouse side, this product, this formulation is used to promote branching and increase flower set. So some of the products it's used on is Christmas cactus, echinaceas hostas. A lot of the perennials are starting to use this early on in order to promote that branching, have more branching, fuller plant. It's going to lead to better flower production. We can use it both in liners and in finished plants. It doesn't readily move within the plant, so the application coverage is going to be key. If you're not getting good coverage with this foliar spray, you can see, it will show itself. You'll see one side of the plant or a couple branches. If they were not covered properly, you're not going to see the branching, and then you'll see that lack or that hole in the flower production. So it's key to make sure you get good coverage, not just with Configure or benzyladenine, but with all of the PGRs. Gibberellins. So we talked about stopping gibberellins, but sometimes we may need gibberellins in order to help promote growth. Okay, so Florgib and ProGib TNO are two of the brands that are out there. Growers can use this if for some reason they over applicated. They put on too high a concentration of a PGR and they need that product. They need to help it to get out of that stunted state. So it can be used in this case to help or ameliorate an over application of PGRs. It does promote stem elongation also in tree form of plants. So, certain lavenders or things like that, sometimes in hibiscus, where they use those little topiary type plants, they'll give them a shot early on in order to elongate those plants, get them to the height that they need, and then grow them out from there. So sometimes we do need gibberellins. And then now there's even a combination of where we've got benzyladenine and gibberellic acid together. And the two brands that are out there are Fresco and Fascination, often used in potted lilies as a foliar spray. And what this helps to do is avoid that lower leaf yellowing and leaf drop that normally occurs. Lilies are a long crop that are in production. So sometimes there's issues with that lower leaf yellowing, and the application of this product, these two actives together, helps to fix that problem of that lower leaf yellowing. It also helps to prolong that flower life, so that flower stays open longer, so the end consumer can enjoy it for a longer period of time. So it's a unique product, but it has a specific niche. So to wind up, some tips for success. Again, make sure you're reading and following the product labels. Be aware of the application methods, whether it's a spray, a drench. There's a lot of research where we're talking about sprenches, where we're talking about dips and things like that. And all of that information is on that product label. If you are applying as a foliar spray or even as a drench, make sure you are putting out that product uniformly. We want to make sure we get good coverage, that we get the foliage covered, because in most cases, especially as a foliar spray, it is being taken up by the leaves, some by the stem, but predominantly by the foliage. Time of application will also affect how well this product works. So if you can apply early morning when light levels are low, or on cloudy days, it's ideal, because the longer that product, the moisture that product can sit on that leaf, it just improves the uptake of the active ingredient. And make sure the leaves are dry. So if it's an outside application, if you're doing perennials outdoors, if there's morning dew on them, you might want to wait until that cools off. So instead of a early morning application, you may have to hold off until end of day when your light levels are going down in order to get that application on. As I mentioned before, more is not always better. So stick to the label, stick to the recommendations, stick to the dosage rates. And as I also mentioned before, these PGRs, the AIs, the active ingredients, on most of these are off patent. So there's many brand names and there's many formulations out there. So just make sure if you switch, if you're used to using Paclobutrazole and you've normally used Piccolo and you're switching to one of the other ones, like Bonsai or Paco or something, check the formulation before you mix up your tank, because you want to make sure, there might be a change in that formulation and that concentration, and you don't want to go based on what you've applied in the past. You want to do it based on the product that you are working with. So with that, Irfan, I am done.
Awesome. Thank you so much,
sharing.
Paul. And from here, we did originally have scheduled a guest speaker, Dr. Vikram Veliga from Texas Tech University. Unfortunately, something else has come up, so he was unable to make it today. But fortunate for you, you get to hear me a little bit, or unfortunate for you, I don't know yet. So let me share on my screen. Do it this way. Here we go.
I do have a quick question about PGRs.
Oh, please. Actually, if there are any questions, not only from Laura, if there are any questions, please put them in the chat or pipe up and yeah, go ahead and ask your questions.
Well, I know that this is nursery and greenhouse day and that you were, and that PGRs are primarily used in floriculture, but do you have any experience using them in the landscape? People are always wanting to try, to regulate the growth of, let's say, a very popular ground cover that requires a lot of string trimming, just a few things like that. Do you have any experience with that or any?
Laura, I have not done any trials, and I'm not aware of any, landscapers that are using it. We had a tree conference here about 2 years ago, and there are some arborists that are using it as part of their pruning, in order to maintain. Yeah. But on ground covers, I haven't come across anything, and I have not done any trials on them in the past, but it sounds like it might be a spot or an area that may need some attention.
I think there's some potential there.
Yep.
Are there any other questions for Paul? Well, if you have any other questions, you can still put them in that chat. There is still time, so feel free to do so. I thought I'd take a moment, since we are starting to get into a little bit later in this season, maybe a little quick review of crop scouting strategies in an IPM program. And I always like to start off by saying, reminding people that When we're talking about insect management, it's like, we're in a battle with little mini aliens. So this is actually an example of a type of fungus, commonly referred to as cordyceps, that has actually infested this ant. Actually alters its neurology, its brain and behavior, causes that ant to climb up on branches, clamp down with its mandibles, and then it sporulates so that those spores can spread onto more ants, right? So it manipulates the ant's behavior to further spread itself, which is, again, it's very alien-like if you think about it. And every once in a while, we see these little specks of dirt. I actually saw one just a couple days ago and took some video footage of it. It looks like a little piece of dirt kind of moving around. And that could be one of two things. Either one is this lacewing larva that's under this debris of the carcasses of its past enemies and meals, and it's using it as a bit of a disguise and protection. Or two, it might be that you're losing your mind. So if it's the second one, do not seek help from an entomologist. Unfortunately, we are not trained to deal with that. But the first one, we are. Lacewing larvae are beneficial predators, so they are things we want to keep around. And that's what scouting is all about, right? Is knowing what are pests and what are our allies, and at what point do we actually require some level of intervention in order to reduce the pest populations. So we thought we had perhaps resolved this back in the 60s. The advent of synthetic insecticides is DDT. You can see powerful insecticide, harmless to humans. And there was 400,000 tons of DDT applied annually worldwide to manage mosquitoes. Now, in its defense, right, it was highly effective. Malaria cases was cut from 8 million cases in 1943 to 800 cases 15 years later. So drastically reduced the number of mosquito populations that were vectoring malaria. And it really eradicated malaria from southern US. However, this is where we also learned about things like environmental toxicology, human toxicology, insecticide resistance, right? If we spray the same thing over and over and over again, that we are essentially selecting for resistant populations. And so in comes integrated pest management, or the pyramid of alien diplomacy, just for fun. Why not? All right. So the very base, the very foundation of it, right, is we want to know who are your enemies, know who are your allies, and know what populations they're at, right? And that's the scouting and monitoring and what we're going to be talking about today. Then we have cultural control practices like habits and best practices, physical and mechanical control. We're talking about barriers or physical removal. Biological control, so that might be like beneficial predators and parasitic wasps, or things, insecticides that are considered softer, so they have less non-target impacts, maybe lower residual, lower environmental impact. And then last resort, as a last resort, if not all of these fail, which we want to continue refining and improving on these things, if they fail, then we want to nuke them. Right, that's when we come out with our big insecticides and we nuke them. Now, while we don't want to resort to this right away, is because in the long term, it's more costly in every way, from environmental impact, human health, but also insecticide resistance and the number of applications you may have to end up doing. So this type of situation here creates an environment that's reliant on human intervention to continue to come and clean up. We call it the pesticide treadmill, where we literally are creating an environment that requires regular insecticide applications. Whereas if we can create a system in which relies on these a little bit more, then we can create something that's a little bit more balanced, or create a slightly more less human reliant system when it comes to the pests that we commonly face. So we're using this monitoring and scouting to make decisions about all of these, about our cultural practices, physical, mechanical, biological, and insecticidal. That's why it's so vital, and that's why this is going to be kind of dedicated on that. And these management decisions are going to be based on thresholds, right? So a certain number of whiteflies per poinsettia, a certain number of chrysanthemum leaves or branches infested with spider mites, so on and so forth, right? So depending on the value of the crop, the damage caused by the pest, right? So if it's actually reducing the marketability of that plant, or is it actually vectoring a pathogen or a virus? So we get into actual monitoring tools. These are things that I usually have on me or very close to me. So whether it be yellow sticky cards, which are highly effective at catching a lot of flying insects, but they're not going to catch either non-flying insects, nor are they going to be very indicative of the actual population in your crop. So don't think that just because you see no flying thrips on there or no winged aphids on there, or very few winged aphids on there, that you don't potentially have a high population of aphids in your crop. Yellow and or blue sticky cards should just be used as indicators of what to scout for on your actual plants. That's very important. Don't use it as, for determining your thresholds. Hand lenses, and we'll see here in a moment that sometimes I prefer to actually use a camera lens. Little contraptions and containers, which are always very useful for actually containing specimens as you're inspecting. Use aspirators for collecting a lot of small insects, especially small winged insects, again, for closer inspection later on. And yellow sticky tape. So this is actually not only used for monitoring, but can also be used for excluding pests from your particular greenhouse as well. Now, there is a bit of a learning curve to identifying things on yellow sticky cards. All right, it is very easy to assume that this is white and fluffy. That's a whitefly, but it's not until we get a little bit closer that it's actually a type of aphid. This is a woolly aphid, which is rarely actually a pest on a lot of those greenhouse ornamentals. So you might mistakenly think it's whiteflies and start spraying for whiteflies when you don't even have them. So very important to train yourself, spend some time, ideally if you have a hand lens, to learn the difference between these pests on a card versus on the plant. And there is, again, they look quite different on the sticky card because they're kind of squished and flattened out. All right, so this is that woolly aphid right there when it's actually standing up. When it's standing up, you can tell much easier that it's definitely not a whitefly. But again, when it's kind of stuck there on the card, it's a little bit different. Whiteflies, when they get stuck on there, their wings get all mangled up, and they're not so white and waxy anymore. Kind of their bodies are what are more apparent, which are a little bit more yellow, and you'll see the wings are just kind of crumpled up. So this is what a whitefly will typically look like on a yellow sticky card. We have different species of whiteflies as well. So this is the banded wing whitefly. Again, not really a common problem in greenhouse ornamentals. Just because you have a population in your sticky cards doesn't mean that they're colonizing and potentially, gonna result in economic loss as a result of their presence, right? So again, important to see that this is an indication that, okay, maybe I should start looking at my plants to see if I have a population. I highly recommend, if you just look up Headband Magnifier on Amazon, you can find all different types. So if you have dedicated scouts or not, I suggest having these handy, because instead of using your hands, using one hand to flip a leaf, the other hand for a hand lens, these are very useful because you can have both of your hands free. So you can have this thing mounted on your head. Use both hands to flip over leaves because those insects usually are on the undersides of leaves. I did mention, usually I don't use a hand lens anymore. I commonly use a camera lens. This is just an example, not an endorsement of one that I know works well, because there are many that do not work well, that will cause a lot of distortion in the image, or actually take the image completely out of focus. You can't refocus it. But this one works quite well and actually comes with a wide angle lens that twists onto a macro lens. So it gives you about 15 times magnification, and it clips onto your phone like this. So you get some good pictures or videos of insects. And I actually have this one, again, not a specific endorsement, but it is one that I know works reasonably well. It's the one that I actually use, and it's a case. You need to buy their specific case that has a part on it to clip on their lens. So that means their lens is a lot smaller and a lot more portable, a lot easier to just throw in your pocket. To have on you at all times. It gives you 10 times magnification and excellent video and image quality as well. And then a microscope. All right, so this is my son here working on a microscope. I have not tested this particular one, so this is not even me saying I know this one works well, but that, just a reasonable light microscope should not run you too expensive. And they are beneficial tools for looking at these insects a little bit closer. And I find sometimes just using my camera lens in front of that eyepiece can get me some very good quality images, high magnification images, again, for identification purposes. Now we have a number of other tools that are, again, not super sophisticated, but the animations are incredible. Look at that. Amazing. All right. So this is we call it the beading technique. It works very well for things like thrips, for example, that usually put themselves in the flowers of plants. And you can tap that flower above like a white piece of foam board. I usually have a white piece of foam board inside a plastic sleeve, so I can use it over and over and over again, and stays pretty well protected. And I'll, you slap that flower, say 5 times over that white foam board, and you can see the thrips much easier on there, moving and writhing around than if they are in the flower. And it's suggested that you just standardize how many taps you do on any given flower, so that, if you're seeing way more thrips on one flower, it's not because you tapped it more rigorously. You're trying to keep some consistency to how you're actually monitoring for those thrips. We also have double-sided sticky tape we frequently use for crepe myrtle bark scale. So if you have crepe myrtles and want to know when those scale are coming out in order to spray them, we have found over 3 years, multiple locations, they're usually coming out between mid-April to beginning of May is kind of the best timing for those crawlers, the most vulnerable stage of scale, because they don't have the white waxy stuff around them yet. And so we use this double-sided sticky tape, and we wrap around that tree and remove it every week and look at it under the microscope, and you can see all the crawlers here in this case. So as those number of crawlers start to increase, we know that we're starting to get an emergence of all the eggs, and that's a good time to start considering any contact insecticides for getting our crawlers. We also have this burlap band, often used for things like caterpillars. So it's like a burlap sack, basically wrapped around the tree, and a string tied around it, and then the top part is basically folded down, so that you can see you're folded down. A caterpillar going up kind of gets caught in there, or sometimes they go in there to hide. So you can use this as a monitoring strategy or as a management strategy if you want to then go and slap that, or just throw them all into a bucket of soapy water or something like that. A similar technique, but perhaps a little bit messier, is to actually put some thick duct tape around the tree. And then you're going to, on that surface of that duct tape, put some kind of sticky glue. And you're trying to not apply the sticky glue directly on the tree, because it can potentially hurt the tree. But we're applying either bug gum mastic barrier, Roxo bug glue, or Tanglefoot. Any of these, as insects trying to go up or down, it will get stuck in that glue and cannot move up or down. Also have things like pitfall traps can help us monitor ground-dwelling insects. For example, this is ground beetles, which are a good predator if we want to know if we have these predators around. We also have pheromone traps. All right, so a lot of insects, how they find each other in the natural environment, right? So if you think about it, it's a massive forest or outdoors. How does a female of one species make sure she attracts the same species male to her in this vast space? Well, they release a sex pheromone. It's a chemical cue off their bodies. They wing it off their bodies. That's actually a molecular composition. It's all these different molecules in different relative concentrations to make almost like a key that is very specific to each species. And chemical ecologists or chemical entomologists, which I consider like the wizards of our field, have been able to isolate a lot of these chemicals and reproduce them. So you can buy a sex pheromone of a lot of different insects and use them for effective monitoring. So here's an example. This is a chemical cue. Now, in this case, is a feeding cue coming through this tube. And as soon as it goes through, you can see this moth gets very excited and would like to feed. And they've actually used this as like a Pavlovian training to train, because honeybees do the same thing. They'll reach out their proboscis and they smell food they want to feed on. And they've been able to use this to train honeybees to detect very small amounts of either drugs or chemical cues of bombs, to be able to train these bees to help be almost like, what do you call them, like drug sniffing bees instead of a drug sniffing dog, right? So you'll have like security officers now with a little bee on a little leash flying around sniffing your luggage. And if it sticks out its tongue, then you know that's probably when you better bolt and start running. Unless you just have a jar of honey in your suitcase. So in terms of pheromone traps, here's an example of one for, this is called field bindweed moth. This little thing right here is actually the pheromone. So it's actually releasing out of this little thing here over time. They usually last about a month. And you can see here, these are all male field bindweed moths. Okay, so it attracts all the males. You know, that's when they're getting ready to mate. So that's when the females are laying eggs. So that's when you want to do your spray applications to hit those young larvae before they become much bigger and harder to control. We have them for greenhouse pests too. So this is the sweet potato weevil. In many counties in Texas, we do have a quarantine for ipomoeas. You can't move ipomoeas out of those particular counties because of this specific pest, this sweet potato weevil. And how they determine if it's there or not is through these pheromone traps. And this is an example of a greenhouse grower that wanted to have that quarantine lifted from their county. So what we have to do is get 2 years of traps without any sweet potato weevils to prove that they're not there anymore. And within the first week, we caught them. So unfortunately, that quarantine was not lifted, but again, demonstrates how effective these traps are. There are several different pheromone traps in the market. Nantucket pine tip moth, peach twig borer, just to name a few, beet armyworm. Some invasives to watch out for that might be very relevant is the European pepper moth. This is one that we're starting to do a little bit more work on in the AgriLife Center in Overton. Let me just fly through those. Here we go. All right, we also have some general traps. So this is an example of one for catching beetles. And you know what, with a lot of stressed trees from this last winter, this winter Uri storm that kind of passed, it may be worth kind of monitoring for some of these beetles because their populations are going to do much better than previous years because a lot of them tend to be secondary pests. And so as the trees are stressed, these beetle populations are going to do very well, and we're going to see a greater abundance of them. And so, here's an example of just a 2-liter bottle, a bottle of soda, which is inverted. And we have ethanol here, 70% ethanol, like rubbing alcohol, for example, would work, or soapy water. And you'll see an example where you can use soapy water here at the bottom and an ethanol lure here. So there's a slightly more sophisticated trap so that you can actually incorporate a lure so you're not using so much rubbing alcohol. So you're still using that as an attractant, and then you're using the soapy water to actually capture those beetles. And ethanol is a general purpose lure because, especially for a lot of bark beetles, because ethanol is one of those chemical cues that a lot of stressed trees actually naturally release. And so, so by using that ethanol, we're attracting these beetles that are attracted to stressed trees into our trap. So I'd like to go over kind of quickly monitoring some of these main culprits that we might see kind of in greenhouse and or some of the outdoor nursery production as well. So first, with aphids, we know they are a sucking insect pest. All right, and here is actually its stylet going in between the cells, in this case, of this particular leaf, and going to be feeding on that the delicious fluids of the plant. So here's the proboscis, the feeding mouth part. So they have not chewing mouth parts, but sucking mouth part. And their distinct feature, their distinct characteristic are these cornicles, or commonly referred to as Ciphunculi, right? So these are communication chimneys. So they actually emit these little chemical cues that go to the tip of these chimneys, and then it releases a chemical to their nearby aphids that they can sense with their antennae. In order to, for example, to release alarm pheromone. So it's a chemical cue that tells all the nearby aphids that, shoot, we're all getting eaten up. We got to run on out of here. And there's like a few main species that we encounter, some of which are the cotton or melon aphid, all right, which are the adult wingless females are between 0.04 to 0.07 inches in length. They're pale yellow, yellow-green to dark blue, so they can vary quite a bit in color depending on temperature. And they have these black or much darker cornicles or Ciphunculi. And those are kind of the main distinct features. You also see here the head, that the forehead you can think of is kind of flat. All right. When we talk about another species, green peach aphid, another very common one is yellowish green cornicles, which match the body. So they're not different color like we saw in the melon or cotton aphid. And there's a bit of an indentation here in the forehead. Okay. So you can see that forehead is slightly different shaped. Now that's only relevant What species you have, I would argue, is only really relevant if you're looking at using biological control. So certain parasitic wasps are going to be very specific to the species of aphid that you have. And that's where it becomes more important to know exactly what species you're dealing with. If you're using insecticides, I don't know of too many examples off the top of my head, at least with aphids, where it's important that you know exactly what species it is that you're spraying for. So that's good news. Now, of course, I think we all know what that's sucking pest damage. We get this honeydew, this sticky solution on the leaves that then can get inoculated by a concoction of molds known as sooty mold. Now, sooty mold itself will not hurt the plant. All right, it will not cause any damage, but it does hurt it aesthetically and can potentially reduce growth and yield because, in a sense, it is reducing how much sun that leaf can actually absorb. Now, going on to thrips, they're in this group known as Thyssanoptera, which means fringed wings. All right, and we have several different species such as greenhouse thrips. We have Western flower thrips, which is one of our more common ones. Chili thrips, when you get Houston and further south, one of them is very common. And in terms of their life cycle, they typically lay their eggs in the soil or leaf litter. Now, you'll notice this one, for example, it's actually on a leaf itself. So this can vary. And then, oh, sorry, that I confused myself here. No, the eggs is usually on or in the plant tissue. All right. And then those first instars are feeding on the actual leaves. And at this point, they're very hard to see with the naked eye. So again, it does really help to have some kind of magnification. You'll see here, there's actually some spider mites and spider mite eggs as well alongside these thrips. Now, that's actually not such a bad thing because Western flower thrips will feed on spider mite eggs and help produce your populations. So if you have just the right balance of the two, that's not such a bad thing. And then when you get in the soil and the leaf litter, is where you're going to get the prepupa and the pupa. Now, this is where they can sometimes happen on the foliage as well. It's not exclusive just to the soil and leaf litter. And they're not feeding at this stage. So if we're talking about insecticidal applications, that's why it's important sometimes to spray on, say, a 7-day interval, because that first application might not get those prepupa and pupa. And then a week later, if you spray again, you can actually get all those ones that emerged as new adults. So you can basically, it's called breaking the life cycle. And they have sucking, rasping mouth parts. They're technically not sucking, sorry, they're actually rasping mouth parts. So scratching at that leaf surface, causing that leaf to bleed out juices, and they'll suck up those juices dry. So this is an example of what that damage can look like. You get this silvering and stippling type damage and discoloration as a result of thrips feeding. And again, monitoring technique, one of the very effective methods is A, look for damage, and then B, using this beading technique to get them on a different surface because it'll be a lot easier to see there. Now, when we're talking poinsettias, as they're coming into season as well, the actual production, one of the common thrips on poinsettias is the poinsettia thrips, or also known as echinothrips, and they are, as adults, black, and as nymphs, rather white. And so they can almost look like the hairs on the bottom side of the poinsettias. So again, doing this beading technique can really help you know what is a plant hair versus some thrips. Now getting on to whiteflies, speaking of poinsettias, right? Whiteflies are one of the most common pests there. We have a couple main species. One is more common than the other. But we have greenhouse whitefly and we have the sweet potato or silver leaf whitefly. And this one here is probably the most common that we actually get on our poinsettias. Now, in terms of the general life cycle, we have eggs that go from these different nymphal stages. Now, at this point, they're relatively transparent and relatively flat on the leaf. So they're very hard to see with the naked eye, and they're already starting to do some feeding at this point. And then they all pupate. So at this point, they kind of inflate quite a bit, and they're pupating into an adult form. They'll come out as these winged adults. They also produce honeydew, and you'll get sooty mold in their high populations as well. Now, in terms of differentiating between species, right, I already talked about the banded wing whitefly that has these bands on the wings. When you're talking about greenhouse whitefly and sweet potato whitefly, they look very similar. And one of the main differences might be the way they hold their wings. So greenhouse whitefly kind of holds it a little bit more flat, whereas the sweet potato whitefly, it's like a very steep roof, right? A type that you don't want to go on. And so a little bit more angled. And another kind of characteristic that people commonly notice is that the exuvias, that's after the adult has come out of the pupa, what's left is this little husk. And on greenhouse whitefly, they're quite long, whereas on sweet potato whitefly, they're rather short. Now, going back here, I say this is complicated. The sweet potato whitefly is complicated in the sense that I keep talking about it as if it's a single species, but more recently it's been discovered that it's actually a species complex, over 20, maybe over 30 actually distinct species. They're genetically distinct. And then after seeing that they're genetically distinct, they found they're reproductively isolated as well. So they actually, even though they look physically almost identical, they have many characteristics that are different, including not being able to procreate with each other. And why that's relevant in this case, right? Like I told you earlier, I don't know if too many examples are in aphids. It's important to know what species you're dealing with. In whiteflies, it's a little bit different because some have much higher resistance, insecticide resistance than others. So here, as an example, these are two different species designations of these whiteflies. We have this MEM1 and MED species. And you'll notice when we're talking about insecticides, pyroproxyfen, pymetrocine, or kynoprene, or commonly referred to as Distance, Endeavor, and an NSTAR, are some examples of trade names they go under. You'll see it, they work against this MEM1, but not at all against MED. And when I say not at all, it's to the point where scientists that are trying to maintain just this MEM1 species, because they can't tell the difference between MEM1 and MED, and they don't know if they have a mixed population, unless they remove a bunch and grind them up and do molecular analyses, they will regularly spray pyroproxyfen, and they know that their population will be only MEM1. They all survive, and it's only MEM1. They can run genetic analysis, and it shows up all MEM1. If you reduce insecticide pressure, you start to get a little bit of a shift from one to the other. So you can actually start to reduce when you reduce your insecticide applications. You can start to get kind of more of a little of a mix of the population. For whiteflies, much easier to monitor, again, using sticky cards. You can see here, this is like all fungus gnats, right? So at this point, it's a good time to replace your card so you can see things a little bit more clearly. And yellow sticky tape, also an effective method for monitoring whiteflies. But again, using that head lens and looking under leaves should not be underestimated. For two-spotted spider mites, okay, they're aptly named because they have two spots, and they are spider mites, and here are their eggs. You get a very high population, you start getting some webbing, so you want to avoid that situation like this. At this point, this is a cull. You want to get rid of this plant, or at least prune off that part of the plant, because there's no insecticide, to my knowledge, that can penetrate that webbing and get you good, effective control against spider mites. Now, they have similar damage to what we saw with thrips and aphids and whiteflies. They cause this discoloration and silvering, and that's when you want to look on the other sides of the leaves a little bit closer for who is the actual culprit. In terms of their general life cycle, get egg, larva, and at this point they have 6 legs, and then we have nymphal stages, which then they have 8 legs. So they're technically not insects. Insects have 6 legs, whereas arachnids have 8. And they go through this first, second stage of nymph, and then they become adults. And that whole life cycle can take anywhere between 8 days to 40 days, depending on temperature. You can see that's a huge variation in time. That's because as that temperature increases, they start to reproduce much quicker. So very important to stay on top of monitoring them as temperatures start to increase. Now, again, signs of them is bronzing, start seeing webbing. That's a very high population. They are affected drastically by the weather. So dry, dusty conditions really promotes their populations. They do much better in those types of conditions. And we want to control them with miticide, sulfur, or insecticidal soap. Now, with insecticidal soap, we always want to be cautious with phytotoxicity. We're talking about high temperatures and humidity. They also do very well in high fertilization. Actually, a lot of these insects do well when we over-fertilize our plants. So that fertilizer, that nitrogen, that can make more amino acids. Those amino acids make more proteins. Those proteins make more babies. And they are just baby making machines. So if we over fertilize, we are promoting their populations. And we want to avoid broad spectrum insecticides. So if an insecticide has a very broad range of insecticides labeled on it, there's a good chance that it might be impacting some of the predators of these spider mites. And again, that is not ideal. I also spoke earlier about how Western flower thrips can help manage spider mites. So there are some cases where having too rigorous of a management program for Western flower thrips can then result in a secondary pest outbreak of two-spotted spider mites. Going on to scale and mealybugs. So both look very similar, right? They are kind of waxy on the outside, both sucking insect pests. Now, mealybugs belong in this group Pseudococidae. So these are Cocidae, all right? These are Pseudococidae, meaning they are kind of fake scale, okay? And the reason why they're kind of fake is because scale, the females, lay eggs all in one little ovisac and she dies in there and that's it, she's done. Whereas with mealybugs, that female can lay multiple egg sacs. So she does not need to just die when she lays that first batch of eggs. So they're sap-feeding insect pests. They can infest all parts of the plant. Now, in general, they do not cause plant mortality. So when we're talking about the landscape, they're not a major concern in general. We'll see an exception to that rule. Now, in greenhouse production or nursery production, they are considered a major aesthetic issue, right? They're very, very easy to spot. And as a result, typically have very low tolerance for their presence. So two main kind of categorizations. When we're talking about scale, we have soft scale, which produces thin, waxy, powdery outer layer, which cannot be separated from the rest of the body. So if you actually tried removing that waxy thing off the tree, it would remove the whole insect. So with crepe myrtle bark scale, if you try to remove that scale off the tree, it removes the whole insect, and they produce honeydew, and as a result, you get sooty mold. Hard armored scale, in contrast, that hard shield-like cover is not attached to the body. So if you remove that cover, that insect is still under and on that tree, and they do not produce honeydew. It's relevant to know the difference between those two because in some insecticides can work effectively against one and not the other. So they're a little bit specific in terms of what insecticides are effective against them. So some examples of soft scale is like the false oleander scale, licanium scale, calico scale, and crepe myrtle bark scale. We're talking about hard or armor scale. Some examples include the T scale. We have Cycad olicaspis scale. Now I mentioned there were some exceptions to scales causing mortality. This is one of them. Cycad olicaspis scale, if not dealt with, will cause mortality to the tree, even in the landscape. So very important to manage them. Unanimous scale. And just a little bit more about the crepe myrtle bark scale. Here is the female egg sac, and this is similar to some other scales. The female egg sac is kind of larger and more round, and that's where all the eggs are going to be as well. Whereas the males, smaller, more oblong, are actually going to pupate. So they're going to go from nymph to a winged adult, and they're going to fly, disperse. And so in terms of general life cycle, you can see here, gravid female, these eggs, nymph, female, again, and repeats. Now those nymphs sometimes become these males. You can see the pupa developing into this winged male. So this is a winged male scale insect. Again, Cycad olicaspis scale can be devastating. So this is one where a systemic drench perhaps might be at least a season-long solution to managing that particular scale and needs to be controlled pretty rigorously. Now, mealybugs, like I mentioned, they are Pseudococidae, right? So they're kind of fake soft scale, again, because they lay multiple egg sacs, okay? And they emerge as these crawlers and nymphs. We have several species, such as long-tailed mealybug, citrus mealybug, and pink hibiscus mealybug. Here's an example, again, of their eggs. You can see some of their immatures and some of them starting to mature here. Now, in some cases, we might see things that look like mealy bugs, but are actually not. So this is an example of one known as a Mealy Bug Destroyer. So it's actually a type of a lady beetle larva, often called a ladybug larva. It's technically a beetle. So it'll actually look for a lot of soft-bodied insects and feed on them. We have some species here that look very similar to this, that are lady beetles that will feed on, say, crepe myrtle bark scale. And so they are actually beneficial in the landscape and also potentially beneficial in the greenhouse as well to have around. Now, mealybugs, I've seen this type of situation before. If you are reusing pots, be very cautious. If you've had a high infestation of mealybugs before, they can stick around. So next time you go to pot plants up again, you can starting with a mealybug population. So make sure you do proper sanitation, or if you can't do proper sanitation, then maybe don't use those pots again because you're going to run into some major issues there. All right, now flea beetles, they're aptly named because as you approach them, they quickly flee. All right, they will hop away and hard to see, and yet their damage is quite obvious. Here is what their eggs look like. They're kind of a little bit flat and laid in clutches. Here's their larva. If you've ever seen lady beetle larvae, they almost look like lady beetle larvae. Or a lot like any other type of beetle larva in general, right? They have three jointed legs near the front. They have a distinct head as well. And you can see over time, now they start to become like a big piece of snot, right? So you have basically a snot on your leaf, starting to chew at it. And then over time, you start to get some actual damage from the adults. So this is the water primrose flea beetle, all right? The potato flea beetle, and you can see they're rather small. We also have the striped flea beetle and the red-headed flea beetle, which is starting to become a much bigger issue, especially in the Northeast. Very interesting to know if they're becoming more of an issue here as well. But a lot of flea beetles, the way they feed is usually very selective. So there will be a bunch of holes all over in your foliage. And that's usually a sign that you might have some flea beetles. It could also be a sign of young caterpillar larvae because they're not so much selective as unable to chew through harder tissue. So they can't feed through thicker leaf veins. They're feeding on all the soft tissue between thicker leaf veins. So here's an example, almost looks like a buckshot or bug shot that just kind of went through all those leaves, right? So you got all these holes everywhere. Here's an example as well of some more damage. All right, so now on to the last two kind of general categories are bark beetles and ambrosia beetles. And I want to touch on these because, again, with that winter storm Uri, we're seeing, or at least I'm getting more reports of these particular types of pests. Now, the difference between these two general categories is that bark beetles, they tunnel primarily in the cambium just beneath the bark, whereas ambrosia beetles are bore at least several inches deep into the wood. So they go pretty deep into the wood. The bark beetles feed mostly on plant tissue, whereas the ambrosia beetles actually feed on a fungi, which develops on the wood from spores introduced by the actual beetle body. So as they go in, the fungi on their bodies actually infects the tree, and then they feed on the spores of that fungi. The bark beetles, the tunnels are at least partially packed with excrement, their poop, whereas ambrosia beetles, the tunnels are kept open. We'll see in a moment what they do with that frass. The frass is mostly pink or reddish, the color of cambial tissue, whereas ambrosia beetle, that frass is mostly whitish, the color of the sapwood or heartwood. And lastly, these bark beetles, after pupating, each new adult chews an individual emergence hole in the bark, whereas with ambrosia beetles, new adults exit trees using tunnels created by their parents. So relatively few bark holes per beetle. So Ips beetle, as an example, is a type of a bark beetle. They prefer fresh debris from logging, construction activity, or natural events. So again, we're going to see a lot more of these Ips and graver beetles as a result of this winter storm Uri. So living trees can be targets during outbreaks, especially if they're stressed. And it's kind of neat what happens here. So this center right here is actually where the male goes to attract females. It's called the Nuptial Chamber, right? It's where he's doing this like little sexy dance to try to attract some females. and there'll be multiple females that come in there and mate. And each of these tunnels is actually a female. And as she goes through that tunnel, she will lay eggs. And those larvae will emerge and they will create their own set of tunnels. So you can actually know exactly how many mates there were and how many progeny were produced just by looking at that wood and looking at the pattern of emergence. All right, we have a number of different types of IPS engraver beetles that we can kind of watch out for, but ultimately, if you have them, it's because essentially they are a secondary pest outbreak. They're kind of helping cull some of the stressed trees in a way. And there are some that will actually go after completely healthy trees. Now, to my knowledge, we don't have any that will quite do that. But we have, for example, there's the mountain pine beetle that just has completely wiped out a whole lot of pine trees. And you can see this blue stained fungi as a result of that inoculation. And you can see this boring dust. Okay, so this is kind of them as they come out. Some of that dust comes out that they were boring out of. Now, if you see these pitch tubes, that's usually a good sign. That means that, I mean, kind of a good sign, right? It's a sign that your tree is getting attacked by some beetles, but that the plant defenses are pushing them out. So this is actually a way the plant defends itself against these beetles and pushes out some very thick sap, which will harden and capture the beetle inside. And then in 2,000 years, we can extract that DNA and reproduce all of those insects in a movie called the more recent Jurassic Park. Not as exciting. So in terms of their generation time throughout the year, we don't have a whole lot of recent data on it, but we can have larvae in those trees throughout the entire year. They're usually pupating between June and September. We have adults that can be flying throughout the entire year as well, but also laying eggs anywhere between April and usually till September. So pretty wide timing and range. Now we can use these traps, these Lindgren style funnel traps, to capture flying adults. And again, that gives us a better idea of when those populations are out flying and mating, and we can do some preventative applications if we think our stressed trees have a chance at recovery. And so by doing this, we might be reducing those secondary pest outbreaks that might be just helping the tree decline and instead might help with that plant to actually make it. Now, I'm talking about the Asian granulate Ambrosia beetle. Here's the granulate, and I think, what did I keep calling it? IPS engraver beetle. Yeah, so granulate Ambrosia beetles. You can see here the egg and different larval stages, and you can see the adults. In terms of their general life cycle, majority of attacks when they're actually attacking trees is usually between April to June. And then from there, you get eggs that become adults, the more eggs that become more adults. You have multiple generations after that. And the adults overwinter in the gallery, so they'll overwinter in the actual trees. Now, one distinct characteristic of these ambrosia beetles are these toothpicks. And I've been receiving a number of photos of these toothpicks on crepe myrtles, which is super rare. I have not, at least in my experience, I've never seen them on crepe myrtles before until this year. And I think it's directly related, that this would be my guess, directly related to Winter Storm Uri. Because a lot of crepe myrtles I know in production were hit pretty hard and stressed pretty good. And so that would make it just the right conditions for these ambrosia beetles to come in and take advantage of it. And so here's some examples of, you can see the staining that you get in the wood as a result of their feeding. And also for monitoring, we have those Lindgren funnel traps that can also be effective for monitoring for them, again, so that you can do preventative insecticide applications. If, again, you think your trees can have still a chance of recovery and you want to prevent their further decline from these beetles. And that concludes my presentation. So at this time, I don't know if there are any questions. And while we take any questions, I'm going to try and pull up kind of our little closing slides here. Not seeing anything in the chat.
Nope, not seeing anything.
All right. All right. Well, let me pull this up right here. So as per usual, these video recordings, if you think you missed something or you think it'd be useful for someone to catch into it, are recorded and made available on our YouTube channel. So the Plant Disease Diagnostic Clinic YouTube, there's a playlist, the Chat with Green Aggies playlist. So we have all those videos on there for you to watch if you so wish. You can also scan this QR code and it'll take you right to that playlist. And as per usual, we always ask for your feedback. Please let us know if this presentation was helpful or not, what you'd like to see in the future. Again, these are really help us kind of justify this programming and or help us improve it for the future as well. And then let's see what we got up next is a landscape options for edible landscapes. So we're going to be learning a little bit about some of our favorite edible plants in Texas landscapes next week. So make sure you tune in for that.
See you next
Let's
week.
see. Yeah. Does anyone have anything else to add?
Nope, that was a great job off the cuff, being flexible.
Thanks.
That's good job.
I appreciate it.
Stefan's really got a lot of information to share.
Mhm.
All right. Well, thank you everyone for tuning in this week for another Chat with Green Aggies. I want to thank all of the Chat with Green Aggie team for being here and putting this together. And we'll see you all next week.
Next week.
All right. Bye-bye.